afternoon, everyone, and thank you for coming. Great to see the strong in-person showing, and also a lot of people who are joining us online. Welcome to the CTSI Distinguished Speaker Series. I'm delighted to introduce today's speaker, and just to provide some context, I'd like to remind everyone here, heart disease is the #1 killer, and most of the cases of heart disease are caused by atherosclerotic cardiovascular disease. And if you were to probably ask leading scientists around the country, who's probably in the first few names you think of as experts in the area, the #1 pathologic process that affects humanity, today's speaker will probably be at the top of that list. So I'm delighted to introduce Dr. Allan Tall. A little bit more about Allan that you may not know. Allan obtained his medical degree from the University of Sydney. This was followed by internal medicine, training and GI training in Boston. Thankfully, he decided to no longer do colonoscopies and EGDs and focus intensively on research. He trained with Donald Small, who had important contributions on the role of cholesterol in gallstones, and shifting gears to try to understand how cholesterol affects atherosclerosis development. And Allan had really important findings that inspired many new therapies in the field. One of them, through working with a collaborative group in Japan, is characterizing the important protein CETP. And we show that in patients and groups of patients who are missing CETP have high HDL and low LDL. That actually inspired a new way of thinking in targeting lipoprotein metabolism and athero. Some of these have led to large clinical trials that are still ongoing, showing even interesting MACE events. Allan has also done incredible work in characterizing the role of inflammation within lesions, as well as cellular, especially immune cell turnover within lesions. And I think he's going to talk about some of these concepts today.
And you hear me? Allan has been, a faculty member and a leader of a division at Columbia since the late, 70s, where he's continued to, sustain, an amazing research program that have trained leading scientists around the country. One of them was actually a distinguished speaker a few years ago here. His former postdoc, Ira Tabas, actually his first postdoc, I believe. And he's won every award you can think of from leading societies, from American Heart Association to European Atherosclerosis, Society. So Allan, we're delighted to welcome you here today, and welcome back to UCLA. Thank you.
Thank you very much, Tamir. That's a very flattering introduction, and I'm honored and delighted to be invited to this seminar series, and so nice to see so many old colleagues and friends and touch base. And I'm reminded again powerfully what a great group of scientists you have working in the area of cardiovascular biology, if you like, the metabolic diseases. A real powerhouse, and what a beautiful campus you have. I walked around again this morning and was delighted to see the beauty of the campus, the trees, the peaceful setting, and think it must be just great to be working here. And so good for you all, and the young people, keep it up. My talk today is about an emerging area, clonal hematopoiesis and its relationship to cardiovascular diseases. And this is from a review that I wrote a couple of years ago with Jose Fuster, just summarizing the overview of clonal hematopoiesis in cardiovascular disease. It's expanded since that time. It now encompasses hypertension as well. But the concept is that mutations that are acquired later in life, somatic mutations in hematopoietic stem cells, promote a survival advantage in those cells, leading to an overrepresentation of mutant cells in the bloodstream. If additional mutations are acquired in oncogenic genes, may eventually lead to leukemia, like acute myeloid leukemia, but much more often remain as single or double mutations that don't get on to develop leukemia, but rather do increase the risk of atherosclerotic cardiovascular disease, thrombotic cardiovascular disease, and also heart failure and adverse outcomes with heart failure. So the major clinical discovery that focused attention on this was made by Sid Jaswell and his colleagues, and reported first in 2014 in a preliminary form, and then confirmed with several larger studies that the mutations in driver genes that were responsible for clonal hematopoiesis were also associated with atherosclerotic cardiovascular disease. And so the most common genetic variant is DNMT3A, followed by TET2, ASXL1, and then a variety of others, including the JAK2 gene. And in this analysis of several population studies and then a meta-analysis, there was roughly a twofold increase in atherosclerotic cardiovascular risk for most of the mutations. And then for one of the mutations, JAK2, an outsized effect with more like 12-fold increase in risk for atherosclerotic cardiovascular disease. And so we've done a lot of work in this area with the JAK2 mutation that, of course, is well known as a founder mutation for the development of myeloproliferative neoplasms, and also had been linked to thrombotic disease, but now was implicated in a CHIP setting to cardiovascular disease. In addition, I'm going to talk about the TET2 mutation, which is a common variant. And JAK2 and TET2 have in common, as it turns out, that they're both increasing macrophage inflammasome activation as an underlying mechanism promoting atherosclerotic cardiovascular risk. A key early study was done by Ken Walsh and Jose Fuster, in which they developed a TET2 clonal hematopoiesis model. So this was done by mixing 10 to 20% of mutant bone marrow cells with 80 to 90% of wild-type bone marrow cells, then transplanting those into an LDL receptor knockout mouse. So the LDL receptor knockout mouse, as you know, develops atherosclerosis when fed a high-fat, high-cholesterol Western diet. And what they found was that there was an increase in atherosclerotic lesions in the TET2 CHIP model. CHIP stands for clonal hematopoiesis of indeterminate potential, as I've been talking about CHIP. And so in the knockout mice, the lesions were larger. And then the other part of the experiment involved giving this compound MCC950, which is a specific inhibitor of NLRP3. It binds directly to the NLRP3 molecule and inhibits NLRP3 inflammasome activation. And so that reversed the impact of the TET2 mutation, bringing it back to the level of the control. You may notice that in the controls, there is actually no significant impact of NLRP3 inflammasome activation in the process, in contradistinction to some early high-profile reports that claimed a major effect in the standard LDL receptor knockout model. Another important series of experiments on a clinical level have implicated inflammatory processes in the development of atherosclerotic cardiovascular disease. And I think you're familiar with these. The most important single one of these is probably the CANTOS study carried out with canakinumab, an IL-1 beta receptor IL-1 beta antibody, which showed a reduction in cardiovascular endpoints in people receiving the IL-1 beta antibodies. But in addition to that, two colchicine trials showed a reduction in the major cardiovascular endpoint, leading the FDA to fairly recently approve the use of colchicine to prevent atherosclerotic cardiovascular disease in people who'd had an event or who are at high risk. Colchicine probably has a variety of anti-inflammatory properties, but one important one is that it prevents the assembly of the NLRP3 inflammasome on microtubules. So this suggested also a link to inflammasome activation. I learned last night at dinner that a clinical trial has been reported in the media in which they failed to find an effect of colchicine on cardiovascular endpoint in people with ST segment elevation MI that hasn't yet been published, and so it'll be interesting to look at the details of that. But this may still be a somewhat controversial area. And so these two areas became conjoined through clonal hematopoiesis. So the CHIP studies and the studies with colchicine have come together, and also with the IL-1 beta antibodies. And so these studies have together indicated in humans a prominent role of the NLRP3 inflammasome and IL-1 beta, particularly in TET2 CHIP. So one of these studies involved a post hoc analysis of the CANTOS study, in which it showed that IL-1 beta antibodies only significantly reduced CAD in subjects with a TET2 mutation. So it's a little bit hard to believe that, since they were actually a minority of the overall population. And it may be a statistical fluke based on the limited power of the analysis, which was only done in 1,000 people. But it still suggests that there may be an outsized benefit in TET2 CHIP of inhibiting IL-1 beta. And very recently in European Heart Journal, Jose Fuster and Pradeep Natarayan published that colchicine improves atherosclerosis in a TET2 CHIP mouse model, and also in the UK Biobank, people who happened to be taking colchicine and also had TET2 mutations had a significant reduction in cardiovascular risk compared to people either who were not taking colchicine or who did not have TET2 mutations. So again, not a controlled clinical trial, but suggestive of a particular benefit. Mustafa Yeltsin-Kaya in my lab did an experiment using isogenic human iPS cell-derived macrophages, wild type or TET2 mutant, to show that indeed in human macrophages there is an activation of the NLRP3 inflammasome by TET2 deficiency. And I'll talk more about the underlying mechanisms of that effect towards the end. In another line of investigation, we decided to look at a CHIP mouse model using the JAK2. And in this, we mixed 20% JAK2 bone marrow cells with 80% wild-type bone marrow cells. We transplanted into the LDL receptor knockout mouse. We found an acceleration of atherosclerosis in the JAK2 chimeric mice, and just shown here in terms of lesion area. And then we discovered that not the NLRP3 inflammasome, but the AIM2 inflammasome was rather implicated in that when we crossed the JAK2 with an AIM2 knockout and did the transplantation, there was a reduction in atherosclerosis, and also in lesion area and necrotic core area. Whereas when we did this with NLRP3 deficient cross, the effect was not significant. So also in cell culture studies, the JAK2 mutation showed increased activation of the AIM2 inflammasome. And so let me talk a little bit about the AIM2 inflammasome. AIM2 is a cytosolic molecule, and it's activated by strands of double-stranded DNA. And so they may be derived from microbes, from neutrophil extracellular traps. But in addition, oxidative DNA damage or DNA replication stress may be providing fragments of DNA that activate the AIM2 inflammasome. In our model, these two processes, oxidative DNA damage and DNA replication stress, were present. And so we proposed that they might be at least part of the underlying mechanism. So this is from Trevor Fiddler's paper, in which we first described the role of the AIM2 inflammasome in JAK2 CHIP. And so the data suggested that increased glucose metabolism was generating reactive oxygen species in mitochondria that was then inducing DNA damage. Also, the JAK2 cells were over-proliferating in the atherosclerotic plot. They showed increased Ki67 staining and other features of increased proliferation. So the DNA damage then was activating the AIM2 inflammasome, we thought, leading to cleavage of gasdermin D, forming pores that released IL-1 beta and IL-18. And then the cells would undergo pyroptosis, promoting necrotic core formation. And IL-1 beta, we provided evidence was involved in cap thinning. In addition, there was this like cell autonomous feedback involving IL-1 and the IL-1 receptor that was promoting, further promoting cell proliferation. We collaborated with Pradeep Natarajan and Yi Zhu at the MGH Broad, and they devised a method where they would look at SNPs around different inflammatory genes to see how expression quantitative prey loci around these genes would affect the outcome between CHIP and cardiovascular disease. So this is a sort of complicated genetic thing. I'm sure Jake gets it, but maybe not all of us, the rest of us do. But the idea is the genetic prediction of the level of expression of these genes, how does it modify the relationship between these different CHIP variants in atherosclerotic cardiovascular disease? And so they found that AIM2, SNPs that affect the expression of AIM2 modify the risk of JAK2 clonal hematopoiesis, so that predicted increased expression of AIM2 is associated with increased risk of JAK2 CHIP. So that was nice because our mouse model predicted that, and mice don't always translate, as you know, but it seems like this one did. And then a surprise was that for ASXL1 CHIP, the relationship was also found, and Trevor Fiedler used ASXL1 mutant macrophages to actually show that they were also susceptible to AIM2 inflammasome activity. And then another finding was that the predicted expression level of the interferon gamma receptor 1 also was predictive of an adverse outcome of JAK2 and cardiovascular disease. And this was interesting and potentially important because type 1 and type 2 interferons have been shown by us and by others to regulate the expression of AIM2. So this was sort of like mechanistically, internally consistent. Recently, Brian Hardaway, a graduate student in my group, decided to look at the impact of the JAK2 CHIP variant in an atherosclerosis regression model. And so this is sort of a fairly standard thing that people do to look at atherosclerosis regression that has somewhat different characteristics to progression. The genes and processes involved overlap, but some of them appear to be distinctive. And so this was done by the same way as the progression studies had been done, in which we mixed 20% mutant bone marrow cells with 80% wild type. We did the transplant. The mice were on the Western diet for 16 weeks, and then the atherogenic diet was stopped, and they were given LDL receptor virus by adenovirus by injection, and then continued on chow for 5 additional weeks. So the hypothesis of these studies was that the JAK2 mutation would continue to promote inflammasome activation, inflammatory processes, sort of autonomously from a cholesterol effect, and we'd see much more atherosclerosis, and that turned out not to be the case. So in this study, the LDL lowering conditions were not optimal. So, but we got down to a cholesterol level from like 1,000 to down to about 250 milligrams per deciliter. So this is like pretty successful. And then at the baseline, the JAK2 progression mice had much more atherosclerosis than the controls, but there was no significant acceleration of atherosclerosis in either group. It was a little bit more in the JAK2 mice, but nothing like the baseline effect. So the impact of the mutation appeared to be attenuated once you did LDL lowering. And why would this be the case? Well, Brian analyzed the lesions quite carefully, and he looked at AIM2 expression. So Trevor had shown in his Nature paper that in the JAK2 model, AIM2 expression was increased in macrophages in the JAK2 mutant mice. And that was recapitulated here in the baseline samples. But surprisingly, regression completely reversed the increase in AIM2 expression. So simply lowering LDL cholesterol completely abolished the increase in AIM2 expression. And then sort of the penny dropped, and we did a very simple experiment we should have done a long time ago, in which we fed wild type or JAK2 macrophages with cholesterol in a modified LDL preparation. For the aficionados here, acetylated LDL was used because that's a very efficient way to load macrophages with cholesterol. And lo and behold, both in the controls and the JAK2 mice, we got like a 10-fold induction of IL-1 beta release in response to poly-D8/DT, which is used to activate the AIM2 inflammasome. And this is associated with an increase in AIM2 expression. We're still looking to see what underlies that, whether it's an increase in interferons being released endogenously by the cells. But there's a very dramatic cholesterol effect on AIM2 expression, which has not been previously appreciated. I mentioned that the JAK2 macrophages were proliferating more in lesions, so it wasn't quite significant compared to the baseline, but strongly up. And then in regression, the increased proliferation of macrophages was completely gone away. And that was paralleled by a decrease in DNA damage response measured by phosphorylated gamma H2AX staining cells per section. So DNA proliferation and DNA damage responses also were reversed simply by lowering cholesterol. And in contrast to that, and I'm not showing this, 8-hydroxy-DG oxidized DNA was unchanged in these experiments. So we've walked away from that idea, and this is the way we're thinking about it now. So modified LDL, which in plaque would be presumably minimally oxidized or partially oxidized LDL and aggregated LDL, gets taken up probably predominantly via scavenger receptor A and CD36. There is an impact on AIM2 expression, whether this is mediated directly or through an effect on interferons is something we're looking into. In addition to that, the JAK2 variant itself is inducing a proliferative response, but that interacts with cholesterol loading, which gives rise to double-stranded DNA breaks, providing the substrate, which along with increased AIM2 expression is driving AIM2 inflammasome activation and gas-free cleavage. And I didn't show you this data, but we characterized an antibody that recognizes the cleaved form of GAS-D, so you can actually monitor inflammasome activation as opposed to just the priming effect. And it's reversed in macrophages by cholesterol lowering. So, okay. Another chapter in the JAK2 chip mutation story. So Trevor noticed that a highly reproducible effect of inhibiting the inflammasome with caspase-1 11 knockout, gasdermin D knockout, and anakinra to antagonize IL-1 alpha or beta or IL-1 beta antibodies. Shown in the blue bar is an increase in the cap thickness of the atherosclerotic plaque. So this is thought to be the feature of atherosclerotic plaque stabilization, and there's pretty nice data in humans using optical coherence tomography to show that interventions like statins and PCSK9 that benefit clinical outcomes double the thickness of the atherosclerotic plaque. And so this seems to be a pretty good indicator. And the mice don't rupture their plaques, but we can measure the thickness of the fibrous cap over the atherosclerotic plaque. And so in the blue, you see that it's increased by all of these interventions. And again, in the control mice, there's no impact. So you need an additional factor to get true inflammasome activation going on. We collaborated with Lars Magdefessel and the Munich Vascular Biology and the MV Munich Vascular Biobank to look in human carotid atherectomy specimens. Lars showed that in microdissected human atherosclerotic plaques, the rupture, there was a several-fold increase in the expression of IL-1 beta mRNA in cells in the cap region, linking inflammasome activation to plaque rupture, destabilization and actual rupture. And then in their single-cell analysis, they found that IL-1 beta was predominantly expressed in monocytes and inflammatory macrophages. And in contrast to that, the receptor was predominantly expressed in endothelial cells or fibroblasts. There is some expression in macrophages as well, but the highest expression was actually in the stromal cells. So this suggested there might be signaling from the inflammatory macrophages to the stromal cells. And so we collaborated with Murdoch Riley and his group to look at the impact of IL-1 beta in the inflammasome activation model with chip, either with JAK2 or TET2 chip in this case. And this is showing the JAK2 chip data. And then we did the transplantation into mice that had a GFP marker turned on by MyH11 to mark the smooth muscle cells. And our hypothesis was that the antibodies would be increasing smooth muscle cells in the cap region. So we then sorted the cells by flow into green positive and green negative cells, and green positive and green negative cells, and then carried out single cell RNA-seq. I'm not going to show you all the single cell data in detail, but the surprise was that the Allan beta antibody predominantly increased fibroblasts rather than smooth muscle cells. So that was apparent in the single cell data, and also when we stained for green, there was no increase in green cells in the cap of the plaque. It was actually in cells that stained for fibroblast marker. And so this is done using staining for decorin, which is this magenta or purple stain here. And this shows the control with the IgG. And then after giving the Allan beta antibodies, you can see this increase in the cap with the decorin. So decorin is not a highly specific stain for fibroblasts, but it's consistent with the single cell data that indicated an increase in fibroblasts. There it is. Okay, so the data that I've been talking about so far has been done with mice with a high burden, a relatively high burden of CHIP mutations, 10, 20% in that range. What about more common low allele burden variants? What's the impact there? So there's emerging evidence that low allele burden variants may also be impacting atherosclerotic cardiovascular disease. So here is a study done in East Asian cohorts looking at low variable allele frequency, 0.5 to 2% or 2% to 10%, and indicating a significant increase in cardiovascular risk at variable allele frequency from 2 to 10%, shown particularly interestingly for the DNMT3A chip, which turns out overall to have the weakest association with cardiovascular disease, but maybe there's something special about low allele burden, strongly trending for the TET2 mutation, and then combining the data showing a significant effect. And then looking specifically at the JAK2 VF variant, and somewhat surprisingly, a Danish study in a general population in the Copenhagen suburbs found that the JAK2 mutation can be detected in 3 to 4% of the general European population, not selected for being old, using droplet digital PCR. So the variant is much more common than is widely believed. And then moreover, in the same Danish population, they found that JAK2 is present in 11% of people having strokes, thrombotic strokes, and in versus 4% in matched controls. And then also studies from Japan have shown low allele burden JAK2 mutation associated with increased atherothrombotic risk. This is a convenience sample rather than an actual true population-based sample. And then, so we have explored low allele burden JAK2 mutations. So that's like the next chapter that I'm going to tell you about, is in which we mix only 1.5% JAK2 cells with 98.5% wild type cells. And I'm not going to go into this deeply, but the first study that we published on this in Blood fairly recently looked at the low allele burden JAK2 mutation in thrombosis. We found accelerated arterial thrombosis. And the underlying mechanism, as depicted in this editorial that accompanied our paper, was there was an increased production of young activated JAK2 platelets, and they were crosstalking via thromboxane A2 released to the wild type platelets. And so this crosstalk from the mutant, from a few young active mutant platelets to wild type platelets, was driving atherothrombotic thrombotic risk. So this was present in mice on chow diets as well as high cholesterol diets, suggesting, maybe aspirin could be beneficial in this setting. No. We also developed a stroke model, and this is an ongoing study. This is a middle cerebral artery occlusion model, which then gets a partial occlusion, then gets a thrombus, and sort of maybe as expected, but a nice increase in on the Western diet for JAK2, there was a significant increase in infarct volume, and this is shown in absolute terms, and then also as a percent of total brain volume. And you can see the infarct over here. A very nice result. And we're looking now into the mechanisms, whether it involves NETosis or other processes. So this is in the early stages, but I just wanted to show you the breadth and the idea that low allele burden JAK2 mutations may actually be increasing risk of MI and stroke. And then I wanted to delve into somewhat deeply into the atherosclerosis story with the low allele burden mouse that is in press in JCI and should be online in a week or two. And so again, as I mentioned, we used 1.5% JAK2 mutant cells mixed with 98.5% wild type cells. And then we transplanted into LDL receptor knockout mice. And then we found that there was a nice acceleration of lesion area, necrotic core area, and a decrease in cap thickness in this model. And I should mention that in humans with a JAK2 mutation and in mice with high allele burden, 20% JAK2, there's an LDL lowering effect, a very prominent LDL lowering effect. And that's probably related to expansion of myeloid cells in the spleen that are actually eating up LDL. And in this low allele burden model, there's no cholesterol lowering effect at all. So, you know, as you go from lower to higher allele burden, the impact may be attenuated by the fact LDL is going down at the same time. So in this model, the LDL doesn't go down, and there's a nice atherosclerosis phenotype. So we're interested to see if there was crosstalk going on from the low burden mutant cells to the wild type cells. And so we mixed the wild 1.5% wild type cells with 98.5% IL-1 receptor knockout wild type cells. And so what happens is we have basically a reversal of the phenotype for the lesion area and the necrotic core area. It's only partial for the cap thickness, so there's something else going on, which we think is crosstalk of the IL-1 directly to the stromal cells, and we're looking into that now. But the phenotype of lesion area and necrotic core area reversed by having IL-1 receptor knockout, indicating a key role of IL-1 signaling from the mutant cells to the wild type cells in the pathogenesis. And without going into all the details, we found that IL-1 beta acts via the IL-1 receptor and ADAM 1017 to promote cleavage of cell surface phagocytic receptors, which are TREM2 and MERTK. And so the signaling here downstream of the IL-1 receptor via P38 MAP kinase leads to ADAM 1017 mediated cleavage. And so there's reduced levels of TREM2 and MERTK in the lesions of the low allele burden knockout mice that's reversed by having IL-1 receptor deficiency. And so we collaborated with Ira Tabus, who developed a non-cleavable form of MERTK and showed that that had benefit in this model. And we were particularly interested in looking at TREM2 because TREM2 has emerged as a potential therapeutic target in Alzheimer's disease because there are TREM2 loss of function variants that are associated with a form of early onset Alzheimer's disease, I believe. And so Denali Therapeutics had developed a TREM2 activating antibody. We were able to get hold of it, and we gave this antibody a 49 or an IgG control to the low allele burden mice, and we found that there was an induction of TREM2 macrophages in amongst CD45 positive cells isolated from plaque. And the major phenotype was not an effect on lesion area or necrotic core area. The major effect was on the cap thickness, the cap area. And so that was seen both in the control mice in the blue and particularly in the JAK2 mice where the reduced cap thickness was largely overcome by this TREM2 activating antibody. And so Jesse Williams and a group, Cochin et al. in Europe, have also reported data on 49 stabilizing antibodies, and Jesse also found an increase in cap thickness. and the European group found a decrease in necrotic core. We found a non-significant decrease in necrotic core, so it's sort of consistent, but much less impressive than the cap thickness. So we and Jesse found the same thing there. And then we used staining to suggest that both potentially fibroblasts, and we also had single cell data that goes along with this, and possibly smooth muscle cells were increased by the 49 antibody. And we were interested in the underlying mechanism. So Wenli Liu isolated CD45 positive and then CD11B positive cells from aortas, from atherosclerotic aortas. And she looked at potential mediators of fibrosis. And we actually were guided in this by studies that have been done in human and mouse cirrhosis models. And so we looked at all the potential candidates for mediating fibrogenic responses. And there are a bunch of soft calls for TGF-beta responsive genes or TGF-beta responsive genes. But the standout was PDGFB. And so the 49 antibody was increasing PDGFB in both the controls and the JAK2 mice quite over several fold. And so we decided to look more at this. And we had single cell data, and we carried out something called cell-phone DB4, an analysis to try to look at crosstalk between macrophages and stromal cells. And the outcome of this depends a lot on what the input is. So to some extent, it was biased towards our input because, you know, we put in the things we were interested in. What we found is that using the JAK2 model, comparing the IgG control and the 49 treated mice, the major difference predicted by this cell phone algorithm was signaling between PDGF-B in macrophages and PDGF receptor A or B in fibroblasts in particular. And so that turned out to be the major signal predictor. And so to try to confirm this, we used a specific, well, first of all, we showed in the plots that PDGF-B was increased in macrophages. So you have the yellow stain here is indicating the coalescence of the signal for PDGF-B and TREM2. And so that's increased in the JAK2 mice treated with 49 compared to the control. It's also increased in the controls by 49, less spectacular than in the JAK2, and here it's quantified over here. So PDGF expression on a protein level is greatly increased in the plaques in the macrophages by 49. And then, let me just see. Yeah, there's one slide that I left out here. We also showed that there was an increase in PDGFR8 expressing fibroblasts. So just to summarize this section, the low burden mouse has featured the impact of IL-1 released from wild-type JAK2 macrophages to promote cleavage of these phagocytic receptors, MERTK and TREM2. And in the case of TREM2, we've highlighted the role of PDGF-B signaling to potentially PDGF receptor alpha on fibroblasts and maybe smooth muscle cells in stabilizing the plaque. MERTK has a major effect on the necrotic core area, and the signaling pathways downstream of TREM2 involve SYK and beta-catenin, so they're quite distinct from the signaling pathways downstream of MERTK. And so there are non-overlapping signals going from these two different phagocytic receptors that are promoting features of plaque stabilization. And you know, an entertaining thought would be to make a bispecific antibody targeting both MERTK and TREM2 to stabilize both pathways. And if anybody would like to do that, please let me know. We don't have the money to do it, or the skill. So let's finish up with a bit more about NLRP3. I've forgotten what time I started. I have no idea of the time.
Good.
I'm good. Okay. Okay, so much more has been written about the NLRP3 in inflammasome and atherosclerosis. than about AIM2. And so this may be somewhat familiar, but the idea is that the NLRP3 inflammasome is activated by dual signals. The case may also be the same for AIM2. So the first signal is a priming signal that may go through toll receptors, toll-like receptors to an NF-kappa B activation to actually increase expression of the inflammasome components like NLRP3 in particular, and then the substrate pro-IL-1 beta is also an NF-kappa B target. And then a second signal, which is sort of diffuse and poorly understood, comes into play that then leads to the actual activation of the NLRP3 inflammasome, which leads to pro-caspase-1 cleavage. Then active caspase-1 converts pro-IL-1 beta and IL-1 and IL-18 into their mature forms, and then they get secreted and released from the cell. And so in the atherosclerosis field, the early studies by Ike Lutz and Catherine Moore and their colleagues implicated lysosomal damage by cholesterol crystals in atherosclerosis in LDL receptor knockout, standard LDL receptor knockout mice. And then Catherine had a paper subsequently in which she led on from that oxidized LDL uptake by CD36 could nucleate cholesterol crystals in the lysosome. So the emphasis here was on the idea that lysosomal damage and cathepsins were being activated, and cathepsin activation could actually promote cleavage of either pro-caspase-1 or IL-1 beta. I've forgotten, and actually I had a recent conversation with Catherine in which she still thinks that's an important idea, and so it may well be. And then Jose Fuster showed that Jose and Ken showed that in the TET2 chip model, the priming step was increased, and they related this to a change in acetylation at the promoter of the IL-1 beta gene. We recently completed, or Mustafa Yeltsin-Kaya recently completed a study in which he defined a new pathway that leads to increased NLRP3 inflammasome activation in TET2 deficient cells. And let me take you through this. It turned out that there is demethylation of dual-specific phosphatases 6 and 10 in TET2 deficient cells. It's certain for 6 and maybe less clear for 10. Dual-specific phosphatases then act to dephosphorylate junk. And so when the genes are demethylated there's less demethylation. So these genes are inactivated when TET2 is active, and when it's knocked out, they're less active, and so there's increased junk phosphorylation. Others had shown that phosphorylated junk actually can directly phosphorylate a specific site on NLRP3 that then promotes the binding of a D-ubiquitinase complex. The D-ubiquitinase complex contains the D-ubiquitinase BRCC3 and then a scaffolding protein called ABRA1. And then together, this leads to a de-ubiquitination event that doesn't change stabilization, but is a K68 ubiquitin activating event that leads to inflammasome activation and assembly. So this had actually been, this pathway had been discovered in a blind screen for NLRP3 activators or inhibitors. And so it's pretty well documented among several labs. And what Mustafa showed is that in TET2 knockout cells, there's increased activity of this pathway driven by the dual-specific phosphatases being less, and so there's more phospho junk. There's increased ubiquitination of, increased phosphorylation of and de-ubiquitination of NLRP3, which is driving NLRP3 inflammasome activation. So this would be in addition. And then in another series of studies, Mustafa was able to link this to cholesterol loading of cells. And so this will be very, very familiar to the audience here. But when you have loading with modified LDL or knockout of cholesterol efflux transporters, ABCD1, ABCD1, there's accumulation of cholesterol in the accessible pool, and then via Peter Tontono's aster proteins that equilibrates with the endoplasmic reticulum pool. And then actually, as Peter and Ira had shown, then that results in IP3 receptor mediated calcium release and activation of CAM kinase 2. So our contribution was to show that when cholesterol loading occurs via modified LDL or ABCD1G1 knockout, this pathway gets activated and then feeds in to the NLRP3 activation cycle that I showed you earlier. So there's this conversion effect in TET2 cells of the dual-specific phosphatases and cholesterol-loading CAMP kinase 2 to activate this pathway. And so let me show you some of the data. So this is just using ABRO1 knockout mice, a convenient way to show the effects of decreased activity of the BRCC3 complex. And we carried this out using control mice, TET2 plus minus mice, because humans are actually heterozygous for the loss of function. And so this is a more appropriate model. And then the homozygous knockout mouse. And what we found is that there was a reduction in atherosclerotic lesions. This was most apparent actually in the heterozygous knockout because there was less variability in the atherosclerotic lesion data, but with the same trend in the homozygous knockout. And so this just shows the activity of this pathway is increased in the TET2 mutant, and when it's deficient in the ABRO1 knockout, it reverses the effect. And here's the experiment that I dreamed up to look at the impact of cholesterol loading and lysosomal dysfunction. So we loaded macrophages with modified LDL using, again, acetylated LDL to deliver a large load of cholesterol. And we did this in the presence of a Niemann-Pick C1 protein inhibitor, the U compound. This would block exit of cholesterol from lysosomes. We found that this actually increased refractile bodies, which we believe are cholesterol crystals in the lysosomal compartment, and that's quantified over here. So more cholesterol crystals when you load with acetyl LDL and block exit of cholesterol from the lysosome. So what happened to inflammasome activation? So it's completely blocked by the U compound. So here's the control with nice inflammasome activation. It's increased when you knock out ABCA1 and ABCG1. And then when you combine cholesterol loading with the U compound, there's no inflammasome activation at all. And this shown over on the right side is a blot showing the active form of caspase-1 and also the cleaved active form of gastromin D, again, completely reversed. So this experiment indicates that under these conditions, cholesterol must exit the lysosomes to activate inflammasomes. And we did a series of experiments with ASTA knockdowns and CAMP kinase 2 inhibitors, and to show the rest of the pathway that I have shown you. And so the prediction is that there's synergy between cholesterol loading and TET2 mutation. And this is an in vivo experiment to test that idea. We're looking now at splenic monocyte macrophages in TET2 chip mice on chow or Western type diets. And here's junk. This is the phosphorylated form of junk. It's particularly increased by the combination of the Western diet and TET2 deficiency. P20, the active form of caspase, is down here, and it's obviously present when you have TET2 deficiency and the Western diet, and these things are quantified over here. And so only when you have both the Western diet and TET2 deficiency do you actually see significant effects on junk and inflammasome activation, suggesting in vivo significance. Very recently in Nature Medicine was published an analysis of 5 TIMI trials. So the TIMI trials are run out of the Brigham, and they look at cardiovascular medicines in well-designed clinical studies. So they looked at 5 different trials, which included PCSK9 antibody trial and then various antithrombotics. And the follow-up of these studies was limited in time, on average, I think about 3 years. But basically, they found that CHIP was associated with the first cardiovascular event, but not recurrent events. And the presence of CHIP didn't appear to identify patients who would benefit particularly from any particular therapy as used in different trials. But the remarkable thing here was that the risk ratios for CHIP were very low. And the one obvious interpretation, I'll give you another one in a minute, is that these people are extremely well treated. So people in clinical trials get LDL lowering drugs, they get antiplatelet agents, and so their risk factors are very well dealt with. And under those conditions, maybe CHIP doesn't do so much, and particularly after the first event, the therapy is added or doubled, and then you get protection, so you don't get a second event. So that's one interpretation of the data. And the other thing that was remarkable was in most studies, TET2 chip and JAK2 chip have had the most prominent roles in atherosclerotic cardiovascular disease. But TET2 chip was very weak in this study. So maybe what it really indicates is TET2 chip is responsive to, treating risk factors. That's my interpretation. And they did a sub-analysis because one of the reviewers insisted on it. And the adjusted hazard ratio was 1.06, not on statins, and 0.99. So not very impressive numbers, but still maybe in the direction suggesting benefit of statins. So this is the ending of my talk. I'm going to deal with the issue of reverse causation. So there was a paper in Cell in 2021 from Heide et al. Phil Swirsky and Matthias Narendorf were involved in this. And this was a very nice study, which was a theoretical study in part and an experimental study in part. And they showed that increased stem cell proliferation in atherosclerosis is driving expansion, the clonal expansion of blood cells. And they went so far as to suggest that the whole of CHIP association with cardiovascular disease could be explained by reverse causation. The atherosclerotic milieu or disease was actually driving the emergence. So you've got a mutation in a stem cell, and then the atherogenic milieu provides expansion of the stem cells and promotes the process, and that is responsible for the association. And so in some experimental evidence was obtained by looking at the fraction of TET2 deficient cells in the bone marrow and the blood. And when they fed TET2 chip mice with a atherogenic diet, they saw expansion of monocytes and neutrophils in the blood. And the TET2 deficient containing monocytes and neutrophils in the blood compared to the chow diet. And then over in the bone marrow, they saw expansion of the the stem cell fraction as well, being driven by the atherogenic diet. And then a wise voice came into this and said, well, maybe we can have a compromise here, and maybe both things are going on. And so Professor Luce has suggested there may be a vicious cycle in atherosclerosis where the atherosclerotic process was acting presumably by cytokines and converging with other stresses like sleep deprivation and smoking to drive the emergence. But this would, so causation had actually been pretty clearly shown in the mouse experiments, right? You do chip bone marrow transplantation and you get more atherosclerosis. So you can't explain that by reverse causation. But in humans, maybe this was going on with some element of reverse causation. And so this was the compromise idea, and I think a very nice way to put it together. And we've thought about this, and very recently there was a paper from Ross Levine's lab. Ross Levine first showed that TET2 mutations were expanded in white cells, and so sort of discovered clonal hematopoiesis, or confirmed that it was increased by oncogenic mutations in leukemogenic genes. And he did a CRISPR screen using co-cultures of endothelial cells with hematopoietic stem cells and human cells. And he identified a specific deficiency in a histone demethylase called KDM3B, and that it was a genotype-specific vulnerability in IDH2 and TET2 mutant cells. So IDH2 mutant cells have a defect, an acquired defect in TET2. So these are also TET2 deficiency. And this is the histone demethylase. And the interesting thing that we noticed in their paper is that TET2 deficient cells have a decreased chromatin accessibility in the CSF2 receptor gene. So this is the CSF2 receptor is the IL-3 GM-CSF receptor that's very important in driving stem cell and myeloid progenitor proliferation. And then the KDM3B knockdown was taking a low level and reducing it to even low levels. And then so wild type cells were not affected, but TET2 mutant cells died. So this was a specific vulnerability of the TET2 cells. And so this, suggested to us maybe this specific defect is reversed by cholesterol loading. And in fact, we had already shown this. So this is a little notice paper from 2020 by Laurent Yvon Charvet. And he had we actually discovered long ago with Russell Dean that ABC1 loss of function variants were particularly enriched in patients with myelomonocytic leukemia. And Laurent made an ABC1G1 knockout mouse in a TET2 deficiency background and showed that this background and the deficiency of these cholesterol efflux transporters interacted to promote stem cell proliferation, myeloid progenitor proliferation, and accelerated atherosclerosis. And the key signaling molecule was the IL-3 GM-CSF receptor. So this leads to the interesting question of whether cholesterol enrichment from diet or other mutations overcomes the specific defect that TET2 HSCs contain in order to promote myelopoiesis. So that's my last piece of data. To summarize, colon hematopoiesis is a recently identified CVD risk factor that works in broad terms by increasing inflammatory risk, but for every variant, there seems to be a different story to exactly how it's increasing inflammatory risk. Anti-inflammatory therapies may be optimally targeted to patients with CHIP mutations, particularly when they have underlying inflammasome activation, which is the case for TET2, JAK2, and ASXL1 mutants. It is not the case for DNMT3A. It's a different set of inflammatory processes not involving inflammasome debris. CH mutations in cholesterol drive myelopoiesis and activate inflammasomes. Both things are going on. And we wonder whether CH, at least some of the CHIP variants, should be an indication for aggressive LDL lowering. Statins and colchicine, inexpensive drugs, may reduce CVD risk in CHIP. I've mentioned as I went through some of the important contributors to this. Ira Tabus, Marek Wester, Murdoch Riley have been important collaborators. And one of the really nice things about this has been the ability to collaborate with hematology colleagues. And so the convergence between the hematology group and the atherosclerosis thinking groups have been really great. And we really think in completely different ways about this set of problems. So it's been a fertile territory. So thank you very much for listening, and I look forward to your questions.
A few questions.
Thank you, Allan. This may be different for different mutations, but to what degree, or if at all, are the clonal populations overrepresented in lesions or in various parts of the lesion? So you didn't touch upon that.
It is different for the different mutations. So JAK2 are expanded in the lesions compared to the bloodstream. TP53 variants that I didn't talk about also expand in the lesions. They're more proliferative and they expand in the lesions. For JAK2, the expansion in lesions is a twofold process. It represents actually increased proliferation in the lesions, but in addition, leukocytes express integrin receptors at high levels, and so they go more into the plaque. I think Ira told me he thinks DNMT3A is expanding in the lesions. That's unpublished data. TET2 mutants do not expand in the lesions. They expand a lot in the bloodstream, and then they just go in.
This is a microphone. Okay. So thank you for that really elegant talk. I so enjoyed it. So I had a question for you. Just let's say 10 years from now, we have a CHIP clinic. How will we be targeting therapies with the knowledge of CHIP? Will it be an upstream approach like you alluded to, NLRP3 inflammasome, or it will be like a downstream approach you mentioned, canakinumab? Or if we lower cholesterol low enough, early enough, will inflammation be a moot point?
Yeah, I mean, I'm an old cholesterol guy, and so my secret thought is that if you lower cholesterol enough, it may not be a problem, particularly for TET2. DNMT3A may be a different story. JAK2, I also believe that if you lower cholesterol enough, it may not be such a big problem. I think there'll still be some increased thrombotic risk. So I think, targeting the traditional risk factors. And I heard that after this TIMI trial study was published, that some groups decided to close their CHIP clinic because it wasn't really a risk factor, but I think that's the wrong conclusion. Upstream factors, I think NLRP3 inflammasome inhibitors are going to be too immunosuppressive. I don't think inhibiting IL-18, we have some unpublished data that makes us think that it may be quite complicated. Some people think that there are other inflammasomes, so if you inhibit NLRP3, it'll be taken over by other inflammasomes. I think that's sort of like a bit of a naive view of how inflammasomes work. IL-1 beta antibodies, I think they should be tried out in CHIP patients. Broadly, I hope that's being done, actually. I haven't seen anything on clinical trials like GOV. I was involved in a startup that wanted to do that, and they ran out of money, unfortunately. IL-6 is around as a target. I think all of those trials are badly designed. They're ignoring the fact that IL-6 antagonism increases ApoB lipoproteins. What else did you ask about?
I guess I think you answered it.
Yeah. So 10 years from now, I don't know. I mean, I think, yeah, lowering LDL is still going to be with us. I don't know about the rest of it. It's a colchicine. I learned last night at dinner that there's this supposedly negative study. It wasn't negative, actually. It was just showed a neutral effect. But I think if I had TET2 mutation and I had an LDL of 80, I would probably 80 or 90, I would probably take colchicine. If I had a TET2 mutation and had an LDL of 30, I'd like to look for an inflammatory marker to give me a clue, like, I don't know, CRP, but CRP is not elevated in TET2 CHIP. None of the CHIP variants associated with elevated CRP, like, too bad for the universality of that. So there isn't, I discussed with Paul Ricker a couple of weeks ago, there isn't a good inflammatory marker. So how do you tell, whether you've lowered LDL enough and that's the end of it, or whether you still have, so this concept of residual inflammatory risk, I think, needs a biomarker that's better than CRP. Okay.
There's long been a connection between atherosclerosis and Alzheimer's disease via ApoE, and I wondered if CHIP may possibly play a role in some of these neurodegenerative disorders.
There's a Nature Medicine paper from Sid Jaiswal, suggesting that CHIP mutations are protective against that Alzheimer's disease in humans. Nobody's confirmed that in a mouse model yet to establish causation. And there were some things done in that study where they took people out who had disease at baseline. So there's a possibility they were looking at a survivor advantage effect. So in other words, if you had a CHIP mutation and you survive with Alzheimer's disease, it's actually marking other genetic effects that protect you rather than the CHIP mutation itself. But they found an expansion of CHIP mutant cells in microglia in the brain. So that suggests something may be going on, So that's an interesting area. I think a lot of people are actually working on that. We decided not to get into it a bridge too far, yeah.
So maybe just because of time, we'll end here. And if you have further questions, feel free to come up to Allan. I want to thank you again for a wonderful talk, and